Welcome to the series Current Methods in Neurotechnology. This short presentation is about how infrared LED oblique illumination can be utilized to study neuronal circuits in the intact spinal cord, block and brainstem preparations. My name is Szucs and I am an associate professor at the University of Debrecen. Our research group's main interest is to reveal connectivity in neuronal circuits of the spinal dorsal horn. The region is the location of the first synapse between primary efferents, delivering different sensory modalities, and secondary sensory neurons residing in the superficial laminae. Initial processing of the sensory signals, including nociceptive ones, occurs here and result in an output signal that is relayed to supraspinal centers by projection neurons found in the superficial and deeper dorsal horn. The major cell types forming neuronal circuits in the spinal dorsal horn have been described decades ago, but due to the limitation of the classical Golgi technique, most of our knowledge is about their somatodendritic domains. Some of the neurons are restricted to a particular lamina of the dorsal horn of the gray matter, while others span several of these, forming interlaminar connections. The thin outermost layer, lamina 1 or marginal zone, contains a variety of projection neurons that come in different shapes and sizes that are related to their function. Somatodendritic domain of neurons determines their input. Exon morphology will determine their output targets and properties, while intracellular recordings from deep dorsal horn and ventral horn neurons as a byproduct resulted in detailed axonal staining of these neurons. Superficial dorsal horn cells were not readily accessible for sharp electrode studies. Detailed information of the axon morphology of superficial dorsal horn neurons only started to accumulate with the increasing numbers of patch clamp recordings in spinal cord slice preparations in vitro. Connectivity studies, recordings from synaptically connected neurons, however, were still very challenging due to the unavoidable truncation of the neuronal processes and thus gathered information mostly about close contacts. Thus, our present wiring scheme of the spinal dorsal horn mainly concentrate on intrasegmental and interlaminar connections, mostly as a result of the widely used transverse slice preparations. However, the segmental organization of the spinal cord implies that neurons in a particular segment are likely influenced by neurons in neighboring segments. Indeed, early staining studies revealed short and long-range intersegmental propriospinal connections. Furthermore, the spinal dorsal horn not only relays processed information to supraspinal centers, but it is also under the control of several descending pathways that modulate local sensory processing, including nociception. So the question is how to study these connections. Due to the anatomical features of the spinal cord and the trajectory of the ascending and descending axons, there is no optimal slicing orientation. Take projection neurons as an example. Transverse, horizontal, and sagittal slices all severely damage the long projection axons of these neurons. The other important problem is that the dorsal roots carrying peripheral input to the dorsal horn circuitry terminate in an angle spreading like a fan along the whole rostrocaudal length of a particular segment. Slicing will reduce primary efferent input to dorsal horn neurons in an in vitro preparation. An easy solution to this problem would be performing in vivo patch clamp or sharp electrode studies without slicing the spinal cord. These approaches, however, are not very popular and technically very challenging, judging from the low number of studies in the last decades. The thin superficial layers are difficult to target without visual aid. The vertebral column is more difficult to clamp compared to the skull, and even with the spine clamp, the movements of the spinal cord related to breathing within the vertebral canal are substantial. So what if one tries to use the unsliced spinal cord in vitro? The problem is the following. Conventional DIC microscopy uses light that passes through the specimen. When specimen thickness increases, the contrast of the resulting image is gradually reduced as less and less light gets through the tissue block. If the tissue contains lots of myelin, which is typically the case for the spinal cord and brainstem, then details of the surface structures can be lost altogether, reducing visibility to none. A solution that circumvents this problem has been published by Boris Safronov in 2007. The technique described in this paper uses an infrared LED positioned close to the illuminated surface, exploiting reflected light instead of the transmitted light that's used in the case of DIC. Oblique illumination gave excellent results on cut flat surfaces of different preparations from different brain areas, such as the spinal cord, the slide of the spinal cord, or even the whole brain. In this original paper, the author suggested to perform a cut into the preparation to create a flat surface for imaging, and at the same time providing access to recording electrodes for patch clamp experiments. Oblique illumination works well with standard thin slice preparations as well. It gives similar or even better results with a properly positioned LED. The optimal illumination angle of the LED is around 20 to 30 degrees from the horizontal plane. However, good quality images can be obtained in a wide variety of angles, ranging from 0 to 50. 2 years later, the method had been revisited, and we tried using it on the dorsal surface of intact lumbar spinal cord blocks without a cut surface. The dorsal surface of the spinal cord was readily accessible for imaging through the overlying thin white matter. Cells down to lamina 3 could be visualized using animals with the age up to 5 weeks. The condenser of our axioscope microscope was removed completely, and different chambers have been designed to accommodate the different preparations that we've tried. The fact that the spinal cord was not sliced or cut simplified the preparation procedure, the steps of which will be shown now in a short video. To prepare the spinal cord block preparation, you will need the following items: 2 chambers filled with ACSF, a larger one to accommodate the back part of the animal that contains the spinal cord and the vertebral column, and the smaller circular or more shallow container for the removed spinal cord. The surgical tools that you will need to do the dissection will be scissors, spring scissors, fine and coarse forceps, plus preferably a curved forceps. After pinning out the removed and cleaned back part of the animal that contains the vertebral column, within which the spinal cord resides, you carefully cleanse the ventral surface of the vertebral column, removing the aorta and remaining tissue from the ventral surface of the vertebral bodies. This is needed to have proper access for the scissors during the dissection. After cleaning, spring scissor is inserted into the vertebral canal and in an alternating manner, cutting left and right, the vertebral bodies are removed by severing the vertebral arches on both sides. If we need to preserve the dorsal root ganglia, the cut should go as close as possible to the vertebral bodies. Like this, we have access to the ventral surface of the spinal cord, and you can nicely make out the ventral and dorsal roots that run along with the spinal cord, aiming toward its own intervertebral frame. By holding the pia mater with a fine forceps, you can carefully lift the cervical end of the spinal cord while using the fine spring scissor. Again, in an alternating manner, you're severing the dorsal and ventral roots that run towards the intervertebral foramen. Please be very careful not to stretch the cord while turning it from left to right. After cutting all the roots and severing the cauda equina, lifting the spinal cord by the pia mater, you can transfer it to the shallow chamber for further dissection. During this further cleaning, holding the dorsal and ventral roots one by one with the fine forceps, using the spring scissor, you cut these roots away from the spinal cord surface, preferably as close to the entry zone and exit as possible. If you need one of the dorsal roots for stimulating perineal referent input, then obviously you have to maintain and preserve the selected dorsal root. After you're done with the cleaning of the roots, the next step will be to remove the meninges. For removing the meninges, one holds the pia mater with a fine forceps and then peel it off very gently and carefully from the surface of the cord. Since you've cut the roots, the pia mater will usually come off in 4 different parts, and these parts are between the root, entry, and exit zones. At the end of this procedure, you have a clean surface of the spinal cord, and now you cut out the region of interest for the block preparation. We are now cutting out the lumbar enlargement segments from L1 to L6. On the dorsal surface of the cord, the 2 vertical gray bands indicate the dorsal horn. The next step is to fix the spinal cord prep to a gold plate by fast glue. For this procedure, we need to lift out the spinal cord block from the ACSF. This has to be done quickly to minimize the hypoxic damage. Once lifting of the cord, carefully with the filter paper get rid of the moisture from the bottom and drop it gently in the fast glue on the gold plate, and then quickly immerse it again in ACSF. The spinal cord block glued to the metal plate is then transported into the recording chamber, and the setting up of the oblique illumination begins. A regular imaging session will be shown in the following video clip. Adjust the position of the preparation with the forceps, and please make sure that when you're gluing your prep, the recording surface faces upwards. Once locating the preparation with a low power objective, with the objective, and then changing it to higher magnification, you have to lower the objective to be immersed in the solution. Then lifting it gently upwards, you will be able to reach the focal plane in which the surface of your preparation will be visible. At this point, the LED is only roughly positioned, and you'll later adjust it to achieve maximum illumination in the center of the visual field. On the screen, you can see the surface of the spinal cord now, with parallel running fibers from left to right, which is the rostrocaudal orientation in this case. Since this is a three-dimensional structure, not a flat surface, obviously adjusting the using the micrometer screw, one can focus through the preparation. On the roughly position that already gives a good contrast, you can further improve it by setting the three axes of the micromanipulator in a way that it gives maximum illumination in the center of the visual field. Turn the knobs and observe the changing intensity histogram in the bottom right of the image. After setting up optimal illumination, one can search for cells in the visual fields. The structures outlined by the arrow point out on the one superficial neurons in the spinal dorsal horn. As we go towards the midline of the preparation, climbing up on the slopy part, we reach the cut dorsal roots. Then focusing down again in the area where the dorsal gray matter is accessible, one can resolve nice neurons with intact and clear contours. Oblique ER-led illumination can be combined with conventional fluorescent imaging as well. Using an epifluorescent illumination on the surface of the block preparation, one can, of course, visualize fluorescent structures such as these axons tagged with a TD tomato fluorescent protein, a transgenic animal. As you can see, fine details of the labeled axons can be followed with huge precision. As a final step, after selecting the potential candidate cell for patching, one can continue with the descent of the patch pipette, which can be located and visualized the same way as with conventional DIC technique. Once close to the surface, we apply regular positive pressure. Please note the fine caliber and thicker myelinated fibers that are resolved with extreme detail on the surface. As we approach, you can see that these prime reference move away from the tip of the pipette, and you can really nicely distinguish the different caliber fibers close to the surface and the dorsal horn neurons in between them, with sharp, clear, well-defined contours. Oblique infrared LED illumination works well in other brain regions as well, as long as there are neurons close to, in roughly 150 microns to the surface. Ventral horn motor neurons in young animals, where the ventral funiculus is still undeveloped, can be visualized easily. The dorsal surface of the red brainstem also contains regions where neurons can be accessed. The lack of condenser and the freedom of not needing a glass bottom chamber allows the use of various preparations. Two examples are shown here. The first one is a brainstem cervical spinal cord transition zone preparation for studying converging inputs in the trigeminocervical complex. Or, the second example shows, you can perform a brainstem spinal cord U-shape preparation to study spinal descending modulatory connections. These special preparations, however, need special holders and chambers which can be custom designed and printed in 3D from hydrophobic plastic. The technique has been implemented in a lot of different experimental setups. The freedom in preparations is evidenced by the skin spinal cord preparation that was investigated with the ER-led method. In this particular case, the imaging was combined with optogenetics. Another research group recently performed in vivo patch clamp with simultaneous Voltron imaging using an ER-led attached to a standalone objective coupled to a camera. An approach based on our prototype of a simple video microscope combined with ER-led. The main advantage of oblique illumination compared to DIC is that it is cheap and easy to adjust. Using ER-led illumination attached to an objective connected to a camera allows using it in vivo and also a wide variety of in vitro block preparations, making it ideal for studying microcircuits in vitro. It also has to be mentioned that the intact preparations also preserve neuronal structure better, therefore allow more reliable morphological identification. Potential pitfalls of the imaging technique mostly arise from bad positioning of the LED. One has to adjust the three axes of the manipulator holding the LED in a way that light intensity in the center of the visual field is maximum. One also has to avoid contact between ACSF and the LED legs or connector to prevent short-circuiting the LED power source. Another reason to maintain a stable fluid level within the chamber is to avoid changes in the immersed length of the recording pipette, which change capacitive properties. A list of the equipment you will need to set up oblique infrared LED illumination is shown on this slide. An infrared LED is needed, preferably with high intensity, and the wavelength should be selected to fall within the sensitivity range of the camera. A current source to drive the LED is also needed. Battery-based solutions here are a good choice as they do not bring in electrical noise. Their stability, however, is inferior to commercially available proper current sources. The video camera has to have sensitivity in the infrared range, and the imaging software that comes with the camera should offer automatic lookup table adjustment or contrast enhancement. Rolling average or binning may improve the image quality and increase contrast in certain cases, but it is not essential. Custom-made holders can be designed according to your needs with any commercially available freeware 3D modeling software. The ones shown here were done with Blender 3D. Finally, two examples from microscope equipped with infrared LED illumination. The first one is a modified Zeiss axioscope with the condenser and specimen table removed. The LED is positioned using an RSG hydraulic manipulator, which is way too sophisticated for this purpose. The other example is a scientific oscilloscope-based system where a special holder has been designed and printed in 3D to hold the NeurotechEU coarse manipulator that positions the LED. Further details of the technique and detailed description of the applications can be found in these two publications that originally described the technique. Thank you for your attention, and goodbye.